All right, welcome everybody to the July 29th MycoTalks. I'm Jay Kolls, professor at Tulane University, and pleased to co-chair this MycoTalks session with Dr. Oliver Kurzai from the University of Würzburg, Germany. And today we have two great talks, one by Duncan Wilson from the MRC Center for Medical Mycology on micronutrient scavenging by human fungal pathogens, and then a talk from Jules Ene from Institut Pasteur, France, on population heterogeneity in the context of antifungal drug resistance. So the philosophy of MycoTalks is to foster diversity, including career stage, gender, geography, and field of study. We have two 30-minute talks on all aspects of medical mycology for the research community, followed by a 30-minute in-depth discussion. You can submit your questions through the Q&A button at the bottom of your Zoom screen, and we'll field those questions and organize them and ask them to the speakers to address them in the 30-minute discussion period. This session is also recorded and it will be available on the MRC CMM YouTube channel. Our upcoming MycoTalks, we have one in August and one in September. August 28th is Bridget Barker and then Frank van der Verdonk. And then September 30th, we have Carl Kuchler and Kirsten Nielsen. And you have to register for these in advance, and you can register on the website. And also, if you're a prior MycoTalk participant, you will also get an email with the registration link. I've been asked to make this announcement for trainees. There's a new medical mycology trainee seminar series. This is for students, postdocs, clinical trainees, and new faculty. Gives you an opportunity to present your work virtually to the global community. These talks occur on the second Thursday of the month, but timing can be adjusted to fit speaker time zone to encourage international presentations. The goal is to help us highlight the scientifically broad and international medical mycology community by nominating speakers with wide scientific interest in global diversity. You can get in touch and submit your abstract here at this link here. And I believe Jules is also going to put this in the chat so you can retrieve the link from that chat. All right, so, and we also have a new initiative that Dr. Adelia Lars from is going to present, so I will advance the slide. And Adelia, can you present the?
Yeah, thanks, Jay, and hi everyone. Yeah, it's really a pleasure to announce the second seminar series we will be running from the MSc Center of Medical Mycology, and we will start in the first week of September. And this will be called Myco Clinics. And that will be a seminar series in which two clinical experts in medical mycology will be presenting a clinical case study with diagnostic considerations, diagnostic process, treatment deliberations, follow-up and outcomes, and will be concluded with a discussion and overview of the relevant literature. And as you can see, we have found a fantastic organizing team with clinical experts in medical mycology around the globe, and really happy with this team. And we have a fantastic list of speakers lined up to present at those meetings, for which the program is being finalized at the moment. But we just wanted to give you a heads up that the first MycoClinics will be held on Friday, the 3rd of September at 4 o'clock British Summer Time. Jules will provide in the chat the link to sign up for the mailing list, so please be among the first to attend this meeting. But even more important, please let your clinical colleagues know that this is going to be happening very soon, and we hope to see you there. Thank you.
Great, thanks to the very exciting initiatives. With that, I will turn over to my co-chair, Dr. Oliver Kurzai, who will introduce our first speaker.
Yeah, thank you very much, Jay and Adelia, and also welcome. So here I am. Thank you, Jay, and welcome from my side. It's really a huge pleasure to introduce the first speaker, whom I happen to know very well, and this is Duncan Wilson. Duncan did his PhD in Manchester, where he already came into first contact with Candida albicans. And then interestingly, for a postdoc, he came to Germany, to Jena, which is the hub of fungal research in Germany. And this is where we also met for several years. He was a postdoc in the lab of Bernie Huber and detected his interest for micronutrients and Candida. And I remember him talking a lot about zinc. And this is also the topic that he used to establish his own research group back in Aberdeen, in the famous Aberdeen Fungal Group. And then he also switched to Exeter and is now a Wellcome Trust Senior Research Fellow at the MRC in Exeter, and still working on nutrients, micronutrients, and Candida albicans. And I'm really interested in what he's doing at the moment. And Duncan, the stage is yours.
Thank you very much, Oliver. Yes, I'm still working on micronutrients. Just get this opened up. And okay, so yes, thanks, Oliver. I'd just like to start this talk by thanking the organisers of MycoTalks for having me on, and for, sorry, 2 seconds. Technical issues, apologies. Yes, thanks to the organisers of MycoTalks for having me on, and also for organising this whole online webinar thing. It's really, really fantastic. As Oliver said, I'll be talking about micronutrient scavenging in human fungal pathogens. So what are we talking about when we talk about micronutrients in biology? We're really talking about these 4 guys down here: manganese, iron, copper and zinc. And my talk will be focused entirely actually on the micronutrient zinc. And why are micronutrients important to biology? Well, actually, between 1/3 and 1/2 of all proteins interact with a metal cofactor, ranging from central metabolic enzymes, alcohol dehydrogenases, carbonic anhydrases, etc., to important regulators. Of course, zinc finger transcription factors need zinc. And then we have enzymes which pathogens use to deal with other elements of innate immunity. So superoxide dismutases and catalases are both metal-dependent enzymes which pathogens use to detoxify the reactive oxygen species posed by phagocytes, etc. And then in some pathogens, we have classical virulence factors such as zinc-dependent metalloproteases. And why are these such a key element of during infection is due to this interplay here that we call nutritional immunity. Right, just like us, we need to get zinc and iron and other micronutrients in our diet. We eat them, right, in our diets. The microbes that colonize and potentially infect us are the same. They need to get zinc and iron in their diet, but of course their diet is the host tissue itself. And what us mammals have evolved to do is to restrict microbial access to key micronutrients such as zinc in a process called nutritional immunity. And that works very well. That would prevent the growth of many microbes within the normally sterile environments. But of course, these guys here as pathogens are pathogens because these guys can cause disease in the host. So these pathogenic microorganisms, which is Candida albicans, must have evolved countermeasures in order to circumvent nutritional immunity, grow in the infected host and cause disease. Now, this all probably started in the beginning as a kind of like just competition for limited essential nutrients, but it's evolved into quite a complex host pathogen interplay today. And what my group works on is how pathogenic fungi get their zinc. And I use major human fungal pathogen Candida albicans as a model system to study these processes. So this is my talk. I'm really just going to talk about 2 things and try and put them into an evolutionary context. The first is zinc uptake via canonical zinc transporters. And then the second is one of my favorite systems, is the zincophore system, which consists of a secreted PRA1 zinc binding protein and its receptor. So let's start off with the transporters. Now, thankfully, these days we have these excellent resources like FungiDB, and you can basically find every member of your protein family of interest with a couple of clicks to the button. You can take the amino acid sequences and build trees to see how your proteins are related to each other and within the same species and across the fungal kingdom. So that's what I did. Got the zinc transporters from Candida albicans and various other human fungal pathogens. And we see that Candida albicans is pretty neat. It's got two zinc importers. One is called ZRT1, zinc regulated transporter number 1, and the other is called ZRT2. Nice and simple. Okay, how do they work? Well, it's very pH dependent. ZRT1 is effectively switched off at acidic pH and titers on, peaking at pH 7.5. ZRT2, on the other hand, is actually constitutively expressed across during zinc limitation, but it is upregulated under acidic pH. And these expression profiles have a rather predictable impact on growth in Candida albicans. So if we culture Candida albicans in acidic media, where ZRT1 is not expressed and ZRT2 is expressed, of course, the wild type and the ZRT1 mutant grow perfectly well because they're not even switching on the gene. But if we knock out ZRT2, we knock out growth, okay? Whereas if we titer the media pH up to just beyond neutrality, up to pH 7.5, we see this. Both transporters are expressed, so actually knocking out either ZRT1 or ZRT2 does not impact growth in this culture media, because both are expressed, there's redundancy, and we need to knock out both ZRT1 and ZRT2 to block zinc uptake at neutrality. This is mirrored in zinc uptake itself. If we do a zinc uptake assay in acidic media, we see that the wild type Candida albicans takes up all the zinc within about an hour. Knock out ZRT2, we knock out zinc uptake, and in this system we only get partial complementation with the genetic revertant. In neutral alkaline media, story is slightly more complicated. We have almost complete zinc uptake by the wild type within the course of about 3 hours. We knock out either ZRT1 or ZRT2, we get partial decrease in zinc uptake, about a 60% reduction for each. These are the complemented strains here in between. And if we knock out both zinc importers, we of course abolish zinc uptake by Candida albicans. So in Candida albicans, ZRT1 is only expressed and actually only functional, we'll get to that a little bit more at the end, in neutral alkaline environments. ZRT2, although it's upregulated at acidic pH and essential at acidic pH, it can actually support the growth of Candida albicans over a relatively broad pH range. Okay, so that's Candida albicans, that's my model organism, that's the organism that I predominantly work with. What about other fungi? And the take-home message here is that everything is the same, but different. Now, before I started to work on zinc myself, there was a really nice body of literature from the Calera laboratory in Spain, looking at zinc import in the other important human fungal pathogen, Aspergillus fumigatus. And these are just two key papers presented here. Now, what is interesting is if we do a phylogenetic analysis of the Candida albicans transporters that I just described to you, and the Aspergillus fumigatus transporters that Jose worked on himself, we see that Candida albicans, which I just showed you as an acidic adapted zinc importer, is phylogenetically related to Aspergillus fumigatus ZRFB, which is also an acidic adapted zinc importer. So orthologs in two distant related species functioning in the same way, and they're actually regulated in the same way as well. And very similarly, the Candida albicans ZRT1 protein, which I just showed you as a neutral alkaline adapted uptake system, is very closely related to Aspergillus ZRFC, which is also a neutral alkaline adapted zinc importer. So we have orthologous pairs of proteins from two relatively distant related species functioning in virtually the same way. And at this stage, I'd just like to point out that Aspergillus fumigatus has in total 5 zinc importers, because that will become important a few slides down the line. Okay, so what that actually means, this is the fungal kingdom as a whole, and this about third on the diagram, that's the ascomycetes, which is a major major fungal phylum. Aspergillus is over here, and Candida albicans is over here in the two major subphyla of the Ascomycetes. And the fact that they're both operating in the same way, using orthologous proteins at acidic, different orthologous proteins at neutral alkaline pH to take up zinc, means that probably pH-dependent zinc uptake is at least probably commonplace within the Ascomycota, and I would bet money that it is conserved in most Ascomycete species. Right, what about other fungi? As I said, everything's the same but different. These guys down here, I don't work on them directly, but I find them really fascinating. The endemic dimorphs, right? So pretty famous over in America. They're interesting because they cause disease, but they're related to each other, so they're part of the same order. Now, Aspergillus fumigatus is not closely related to these onigenales species, these endemic dimorphs, but it's part of the same subphyla, part of the same order, so it's reasonably closely related. So what we did actually, together with my student Zoe, is we took each of the zinc importers from Aspergillus fumigatus, that's ZRFA, B, C, D, and then this unnamed one, which I'm just going to refer to as ZRFD2 for the moment, because it's quite closely related to ZRFD. So we took each of these and we blasted them independently against the onigenales, the endemic dimorphs. And what we found was this, that everybody has got a copy of the Candida ZRT2, Aspergillus ZRFB acidic. Everybody's got a copy of the Candida ZRT1, Aspergillus ZRFC neutral alkaline. And then up here, we've got this hodgepodge of transporters related to Aspergillus ZRFD, this poorly characterized transporter. So a little bit of a mess. Let's see if we can resolve it. So we did that with reciprocal blast searches. And here's the neutral alkaline, everyone's got one. Here's the acidic, everyone's got one. And then we've identified three distinct zinc transporters present in the onigenales. So those related to Aspergillus ZRFD, those related to that duplicated version of ZRFD, and then a lineage-specific zinc importer present in Paracoxe and Coccidioides, but apparently lost from Histoplasma and Blastomyces. Now, although I don't directly work on these species molecularly yet, I want to tell you that story just to kind of, sorry, I forgot to say, if Bruce Klein and George Deep were in the audience, these are the Blastomyces and Histoplasma transporters that you guys so nicely described and demonstrated a role for in virulence. But the reason I'm showing you this is this reason here. Like me as a Candida albicans person, it's pretty easy to get blinkered on your model organism, particularly when it's got quite a neat two transporter zinc uptake pathways, and just to think that that's the be and end all. But actually, we need to look outside our model to other human fungal pathogens and out with them to the broader fungal kingdom, and see some really interesting diversity into the zinc uptake repertoires of fungi. Okay, so that was zinc importers. I'm now going to switch to the zincophore system. So the zincophore system consists of PRA1 and ZRT1. This is our current understanding of how it works. We have PRA1, secreted protein. It binds zinc via this handsome cysteine histidine motif at its C-terminus, and it associates with the fungal cell via ZRT1, right? That's the zinc importer I talked about, the neutral alkaline zinc importer I talked to you about in the beginning. And it is a zinc importer, so a zip protein, but it's got this large 170 odd amino acid extracellular domain, which itself contains 3 of these cysteine histidine motifs. So that's how we think it is working. And I'm going to talk you through the history of this system. It all started back in Germany 10 years ago now, when I was working with Bernie in Jena in Germany. And me and Francesco, we made this observation. So Candida albicans, we infect human cells. That's the mother yeast cell. They germinate as a hypha, and they invade into the host cell, and the part that has invaded into the human cell is magenta and demarcated with white arrows. Now, we've seen that many times before. And what was interesting was when we counterstained for zinc, this dye called zinquin, we saw the accumulation of what we inferred to be host-derived zinc on the Candida albicans invasive hyphae. Okay, and of course we were interested in understanding the molecular mechanism underlying this. Now, out in the iron world, Siderophores are extremely successful iron capture molecules. So siderophores are they are small molecule iron chelators, which are really good at grabbing iron for the bug. And so we were inspired by that system, and we postulated the existence of a zincophore system. So how would that work? Candida albicans secretes a zinc binding molecule, which binds zinc, gives it to the cell to facilitate zinc scavenging and growth. Now, as I said in the beginning, like between 1/3 and 1/2 of all metals can bind metal. Between 1/3 and 1/2 of all proteins can bind metals, and actually in eukaryotes, a predicted 9 to 10% of all proteins can bind zinc. So we thought that proteins would constitute a pretty attractive candidate for such a proposed zincophore function. So we scanned the Candida albicans secretome, so those proteins known to be secreted, for the presence of predicted zinc binding motifs. And we identified one protein, quite a well-known one since the 1990s, called PRA1, with multiple zinc binding motifs. So we knocked that out and we repeated our zinc scavenging assay. Here's the wild type again, accumulating zinc on the invasive hyphae. We knock out PRA1. Now, importantly, it's still able to invade the host cells. So it still has potential access to intracellular host zinc, as we can see here, and in the quantification on the right, it is severely perturbed at sequestering zinc from host cells. This is a quite an important impact on growth. I'm just going to talk you through this experiment here. We take Candida albicans wild type, or PRA1 delta, we put them onto a plastic surface in cell culture media with no exogenous zinc. Both strains grow very poorly because they're missing an essential micronutrient. It is essential. We supplement with some zinc. They both grow perfectly well, nice healthy microcolony. And if we do the experiment on human monolayers, only the wild type is able to capitalize on that host-derived zinc and grow and form a microcolony. The PRA1 mutant down here, it's blind to the host cells. And it is this zinc acquisition related growth defect which has an impact on pathogenicity. Because when we do a human cell damage assay, in the absence of exogenous zinc, we see that the Candida albicans wild type is able to scavenge zinc, grow, and damage the host cells, and the PRA1 mutant, in the absence of exogenous zinc, is unable to. And then if we supplement the surrounding cell culture media with zinc, the PRA1 mutant damages just fine. So PRA1 itself is not a damaging factor like a candidalysin or a protease or something. It's a zinc scavenger, right? It's involved in getting the zinc for the fungus, facilitating growth, and permitting subsequent damage. Okay, so PRA1 is a protein. It's secreted. It can bind zinc, which I will get back to in the last slide of the talk. But it would be a pretty useless zincophore to the fungus if it was unable to reassociate with the fungal cell. So how might it do that? Now, actually just a couple of years before we started working on PRA1, the group of Aaron Mitchell noted in their PLOS biology paper on ZAP1 that PRA1 and ZRT1 share the same promoter, right? So they're at the same genetic locus. And when we looked at the structure of this zinc transporter, right, with this big extracellular domain, what we predicted is that ZRT1, as well as being a zinc importer, is acting as a PRA1 zincophore receptor, or docking system for the zincophore. So to test this hypothesis, we knocked out the gene and we exposed wild type and ZRT1 deletion mutant cells to recombinant PRA1, and we probed against it, and we see the accumulation of PRA1 on wild type cells, but not on ZRT1 deletion mutant cells. And more recently, Mohammed has made this nice red fluorescent protein. It's an M-cherry PRA1 construct where we can express it endogenously in the fungus. And we see that if we express it in Candida albicans cells, which have ZRT1, we get nice cell surface association. We knock out ZRT1, and that is perturbed. So ZRT1 is facilitating binding and association of PRA1 to the fungal cell. It's the receptor. Okay, as you probably guessed from the beginning with the transporters, although I'm not an evolutionary biologist or anything, I'm quite interested in where these things came from and how they evolved. So what we did is take the PRA1 sequence, we blast it against NCBI. We find no orthologs outside the fungal kingdom, but we do find orthologs throughout the Dicaria. So the Dicaria is the sub-kingdom of fungi, which encompasses the ascomycetes here, and the basidiomycetes here, makes up a lot of species. So we found PRA1 orthologs throughout this subkingdom. in most, but not all species. Now, not only was the gene present, but when we did alignments between Ascomycete, PRA1, and Basidiomycete, PRA1, so these species diverged at least 450 million years ago, probably closer to 700 million years ago, we see high conservation of the zinc binding sites. So the PRA1 gene emerged back here, and we infer from the conservation of the zinc binding sites that its zincophore function is likely the ancestral function of this protein. Okay, not only was the gene and its zinc binding sites conserved, but when we looked at the locus, it was as well. So if we look at the fungal kingdom as a whole, most species, 85% of species that we looked at, have got a PRA1 ortholog, and the vast majority of those don't just have a PRA1 ortholog, but it is synthenic to the ortholog of ZRT1, right? The PRA1 receptor in Candida albicans. So this locus is ancient, right? And we think that is because it facilitates simple transcriptional co-regulation of the zincophore and the zincophore receptor. So common in most, but there's always a but, and it is this. This is how the zincophore is working in Candida albicans. This is the ancestral state of the locus, at least in the dikarya, so the Ascomycetes and Basidiomycetes, last common ancestor, and that has been maintained in most extant fungal species, but a few interesting rearrangements have occurred. So Saccharomyces cerevisiae, this is quite a rare event. I've only observed this in Saccharomyces, and it's actually lost the receptor, but it's come up with a pretty cool way to circumvent that loss of the receptor. It's gained a GPI anchor on the zincophore itself. So it seems to be acting more like Daniel Kornitzer's RBT5 cell wall heme binding proteins than as a kind of typical zincophore. And by the way, in Saccharomyces, it's called zinc and pH regulated surface protein because it's zinc and pH regulated. So pretty sure this is conserved function across most of the fungi. The other rearrangement, relatively rare, I've only observed it in Blastomyces, Magnaportha, Candida guillermondii, and very recently in Candida auris, is where the two genes are still present in the organism's genome, but they've broken apart to distal genomic loci. And then this final rearrangement, if you can call it that, is the wholesale loss of PRA1. Now, obviously, this is relatively rare because I told you that most species have got the gene and most species have got the intact locus. But the first few species that we identify gene loss events in, Candida glabrata, Histoplasma cryptococcus, they're all pathogens. And when we looked at the human fungal pathogens as a whole, these are not necessarily phylogenetically related. We observed independent loss events of the PRA1 gene in multiple, well, actually in the vast majority of the important human fungal pathogens. Now, although the impact of this gene loss is not yet, well, the impact of this gene loss on pathogenicity is not yet known, but we are working on this. It's interesting enough for me to pursue. So it got me interested in what kind of evolutionary drivers might be leading to the loss of the zincophore gene. Now, there is a clue in the name of PRA1. It stands for pH regulated antigen, and the reason for that is because, like its receptor, it is pH regulated. That is the expression of its receptor, and its expression peaks at pH 7.5. Okay, so I took two relatively closely related species, Coccidioides immitis and Histoplasma capsulatum, one of which has kept this zincophore gene, one of which has lost this zincophore gene. They're both fungi endemic to the soil of the United States, and I looked where are they, right? So this is a map of most of the United States of America, and the pink regions denote the acidic soil, right? And white is the neutral or the alkaline soil. So Histoplasma, the species which has lost the neutral alkaline adapted zincophore gene, is restricted to regions of acidic soil, and Coccidioides, which has maintained the neutral alkaline zincophore gene, is restricted to regions of neutral alkaline soil. So what I propose is that because the PRA1 ZRT1 zincophore locus is expressed at neutral alkaline pH, and as I'll get to in the end, functional at neutral alkaline pH, the allele, if not maladaptive in acidic environments, is at the very least useless, and that may be leading to its gene loss in all of these extant fungal pathogens. And you can imagine Cryptococcus in bird guano, you know, it's quite acidic as well. And the take-home message there is that my hypothesis is that ecological pH adaptation is correlating with zincophore loss locus status. Okay, now I talked about alkaline, and I've talked about neutral, and I've talked about acidic environments. What do I mean by that, right? I don't think anybody could argue that pH 4.5 is acidic, but I think it would be perfectly reasonable to also argue that anything below pH 7 is also acidic. So what's an acidic environment as far as the zincophore is concerned? In order to probe this, we developed a new zinc binding assay. That's where we take a zinc responsive fluorophore and we mix it with zinc. And when we mix it with zinc, we get a robust signal. And then the idea is we titer in our zincophore. And if the zincophore has similar or higher affinity for zinc than the zinc responsive fluorophore, it outcompetes the fluorophore, takes the zinc, kills the signal, and we get darkness. Okay. Now, we're now attempting these assays with full-length PRA1 and zincophores from different species and variants, etc. And the data I'm about to show you is just with the predicted zincophore domain here. So the 29 amino acids at the C term of the protein. Okay, so there were basically three major observations we made here when we did the zinc competition assay at different pH values. The first is that above pH 6.8, we get complete zinc binding, right? Although it's actually only really optimal at pH 7.5, but nonetheless 6.8 or above, complete zinc binding. Between pH 6.2 and pH 6.6, we get partial zinc binding by the PRA1 zincophore domain. And then below pH 6.2, even at these supramolar concentrations of the zincophore, zinc binding is negligible. So as far as our zincophore domain is concerned, its threshold acidity is pH 6.2, but its pH optima is 7.5, and this is the gene expression graph, right? So I mean, you know, it's zinc binding optima biochemically peaks at the exact same pH value as its expression. Okay, the reason behind this will be obvious to biochemists, and that's the pKa of histidine. Of course, histidines are the major zinc chelating residues, and their pKa is around 6.3. It depends on the context, right? But what's probably happening is that when we drop the pH to 6.2 or below, all these histidines become protonated, become positively charged, and of course, zinc is a cation, which is positively charged, and we don't get binding. Okay, so this is how the system is working in Candida albicans. We've got the PRA1 zincophore delivering zinc to its receptor, ZRT1. I'd just like to emphasize that the allele, the system and the allele has undergone quite interesting evolutionary trajectories in modern or extant fungal species, so maintained in most, broken in some, and the PRA1 gene lost. Not in that many species, but in a huge number of human fungal pathogens. And now that we've got quite a good hold on the pH optima of the zinc uptake systems, and particularly the fall-off range of the zincophore, what we're hoping to do is take these kind of molecular, genetic, biochemical, and evolutionary findings and try and integrate them with ecological and epidemiological data to see if we can use the zinc uptake repertoire of fungi, so they're like genetic fingerprint, and see if we can use that to predict where in the world they exist. Because, you know, with the exception of some which live within us, most fungal species are out there in the environment. And I'm calling this approach from ions to biomes, because it kind of stretches from zinc all the way up to global habitat ecology. Right, that just leaves me with my conclusion slide. I've talked about transporters, so Candida albicans can take up zinc at acidic pH or at neutral alkaline pH via its ZRT2 or ZRT1 transporter, and the ZRT1 transporter integrates with the secreted zincophore system. But the real kind of take-home message, no matter what fungus you work on, is that we really need to look outside your model of interest. And as far as the major human fungal pathogens go, no single species, no single two species behave in the same way. So Candida albicans has got 2 importers and a zincophore. Aspergillus has got 5 and a zincophore. Histoplasma has got 4, no zincophore. Blastomyces has got 4, like histoplasma, but it's got a zincophore. And cryptococci down here just has the two importers, but has lost the zincophore. So no 2 important human fungal pathogens behave the same. And the take-home message is that when it comes to zinc import in fungi, nobody is playing with the same hand. Okay, so thank you for your attention, and I would just like to thank my group, present, past, and distant past, my funders and my collaborators, and thank you, and I'll pass back to the chairs.
Thank you, Duncan. So just a reminder, we'll take questions after the second talk, and we already have a fair number. We have 7 already in the Q&A. So I'd like to introduce the second speaker, Jules Ene. Jules did her PhD work at the University of Aberdeen with Professor Al Brown, and then did a postdoc with Professor Richard Bennett, followed by research assistant professorship at Brown University, and then now has her own lab at the Institut Pasteur. And we look forward to her talk on genomics and evolutionary genomics in Canada.
Hi, everyone. Can you see my slides okay?
Yes.
Okay. So first, it's really an honor and a pleasure to be part of the series. It's been a real delight listening to these talks in the past year, so congratulations to the organizers. And today I'll try to cover some of the topics that we've been pursuing in our lab and to give a review on our current understanding of how population heterogeneity impacts antifungal drug responses. So I don't need to oversell this topic to this audience, but just very briefly, Candida species are recognized as posing a serious health threat by the CDC. About 7% of Candida bloodstream infections are caused by resistant isolates, and mortality from Candida infections is close to 50%, so much higher than the prevalence of resistance in general. In addition, we have limited therapeutic options. Only 3 classes of antifungals are available to treat systemic Candida infections, and we have documented resistance to literally all of them. Among the antifungals, azoles are the most widely used due to their safety, bioavailability, and wide spectrum of action. And a large part of my talk today will focus on these drugs, which are fungistatic drugs that target the plasma membrane of fungal cells. So in addition, we have hypervirulent multi-drug resistant species emerging, such as Candida auris, which has really become the poster child for how scary these fungi can get, and they have captured our attention with a lot of publicity. However, most Candida infections are still caused by Candida albicans, for which resistance levels have stayed relatively constant in the last few years. However, what we see is increased prevalence of non-Candida albicans species worldwide, including species that are actually intrinsically resistant to some of these drugs, and in particular resistant to azoles. So these include species like Candida glabrata or Candida krusei, as well as Candida auris. So this species was first identified in 2009 and has now spread worldwide and is considered an urgent threat by the CDC. So a third of Candida auris infections are actually lethal, which oftentimes because these isolates tend to be multi-drug resistant. So for example, more than 90% of isolates are resistant to azoles, more than 30% are resistant to amphotericin B, and echinocandins have become the drugs of last resort for the species. So this month, also CDC has reported 2 recent Candida auris outbreaks involving pan-resistant isolates and person-to-person transmission in hospitals in Texas and DC. So not at all good news. So the problem further complicates when isolate persist in infections without being drug resistant. So this is a very hard concept to think about, but we have this paradigm that infections caused by susceptible isolates will be cleared by drugs with a single round of therapy, while those that don't respond to treatment are resistant. And this is still true for a lot of the cases, but frequently we see that susceptible isolates are being treated with multiple rounds of therapy and yet they still persist in the host. So in fact, most fatal fungal infections are caused by susceptible isolates. And in many of these cases, we simply don't know why the treatment fails. And of course, this could be due to a lot of host-specific factors, drug accessibility, but we are particularly interested in understanding the fungal-specific factors that contribute to this type of persistence, because there's mounting evidence now that fungal-specific mechanisms will lead to clinical persistence. So before talking about what we currently know about these cases of susceptible isolates leading to persistent infections, I want to take a step back to revisit how microbes in general respond to drugs. And I'm going to make parallels, draw parallels between bacteria and fungi, because I think we can learn a lot from what others know in bacteria. So first, how can microbes resist stress? So one way to do this is to be resistant. So this is typically caused by a stable genetic change in the genome of bacteria or fungi, which enable the population to grow in the presence of drug. Now, resistant cells give rise to a new population that is genetically identical to the resistant clone when the drug is removed. So in fungi, and I'm mostly referring here to human pathogens, resistance has been described for most drugs and for most species. And we know that this type of resistance is genetic, is stable, and there are a diversity of mechanisms, genetic mechanisms that lead to this type of inherited antifungal resistance in the population. So far things are simple. But they get to become more complicated once we talk about subpopulation phenotypes. So bacterial cells can do this cool thing called persistence. So they can persist as phenotypic variants that can survive treatment, although they cannot grow in the presence of the drug. When treatment ceases, persisters can switch back to the drug-sensitive phenotype and give rise to a new population that is genetically identical to the original one. So this population, again, contains a small fraction of persister cells and a population of susceptible cells. So this is actually very similar to what we know about tolerance in Candida with azole treatment, except that tolerance cells can still grow, although slowly, in the presence of azoles. And the mechanisms enabling tolerance to azoles appear quite different from what's known about bacterial persistence. So finally, much like resistant cells, we can also have heteroresistant cells that can both survive and grow in the presence of antimicrobials. So the key difference here is that these cells tend to be unstable. So they revert to the drug-sensitive phenotype once the antimicrobial drug is removed. So a similar phenomenon has been described in fungi, and I will come back to this. So what I want you to take from this is there are important parallels and distinctions between these phenomena in bacteria and fungi, and it's important to note that we have a lot to learn from the bacterial paradigms in terms of mechanisms and the assays that they do in these species. And a key difference is that most of the work in bacteria has been done on cytal drugs, so drugs that kill the cells rather than inhibit their growth. However, in fungi, a lot of work is actually being done on azoles, and in particular in fluconazole. So these are static drugs, which don't kill the cells, they just arrest growth. And I'm adding a black box on these two phenomena because overall we have a lot more questions than answers about them. But there's growing evidence that for starters, fungal populations are highly heterogeneous. And second, that this heterogeneity can generate specialized cells that can survive antifungal attack. So our drug response is heterogeneous, and how do we assay for this? One easy thing to do in the lab is to use disc diffusion assays. So in this assay, cells are plated as a lawn, and a drug disc is placed in the middle of the plate. And for example, what I'm showing here are examples of one susceptible and one resistant strain, and their profiles on this type of assay. However, most clinical isolates we examine show a lot more diversity than these two strains. And we tend to overclassify isolates as being susceptible despite their diversity in susceptibility profiles. So for example, the first three plates that I'm showing represent isolates, which were all classified as susceptible by MIC. So MIC meaning minimum inhibitory concentration assays, in which the concentration of drug that inhibits 50% of growth is established. However, if we zoom in to these plates, we can see that individual colonies within these populations deal with drug pressures in very different ways. So from those that have slowed down their growth in the presence of this drug disc to those that are completely uninhibited by the drug. And I'm highlighting here and highlighting some of these in red. So this suggests that first we need a better understanding of these intermediate phenotypes, about how they arise, because they could precede the development of true drug resistance. And second, that we're actually missing a lot of the interesting things that these cells do in the presence of these drugs. So a good example of this population heterogeneity in response to drugs is represented by azole tolerance. So this arises due to population heterogeneity. So here, a subpopulation of cells is able to grow at slow rates in the presence of fluconazole. And you can see that this population quite evidently here in the zone of inhibition next to the drug disc. So if we take cells from either the inside or the outside of the zone of inhibition, and we redo this test, we get the exact same susceptibility profile, indicating that this is a heritable and intrinsic property of the population. And we know that we can measure tolerance. We have assays to do this now, and we know that this property is also independent of drug concentration and does not correlate with resistance. So it's an entirely different phenomenon. We also know that multiple stress pathways mediate tolerance. These include HSP90, calcineurin, CRZ1, the TOR pathway, as well as sphingolipid biosynthesis. So this is a complex process that really involves a lot of things going on in the cell. It sounds like we don't know a lot about this, and it's true, we really don't, but we are working on deciphering some of these mechanisms in collaboration with Judy Berman. And here I want to strongly recommend watching Judy Berman's talk in this series for a more in-depth perspective on azole tolerance and how are we defining these things. So a second way in which we can visualize heterogeneity in the population is in response to azoles. And we can do this by using a fluorescent fluconazole probe developed by Benamu et al. So if we incubate low tolerance isolates with this probe, we see that most cells are actually highly fluorescent, meaning that they accumulate a lot of this probe. If we do this assay with a high tolerant isolate, we see that cells now display a wide diversity of fluorescence levels, from some that accumulate a lot of this probe to ones that really don't seem to have any in them. So because our initial observations come from using strains with different genetic backgrounds, we asked if we could see the same thing in an isogenic background, and to really directly show whether tolerance is correlated in any way with the amount of fluconazole taken up by these cells. So to do this, we took a low tolerance isolate and passaged it in the lab in the presence of fluconazole during multiple rounds of passage. And we were able to obtain several isolates that have similar resistance levels, but significantly higher tolerance. And these plots here show an example of such an isolate, and you can see that this isolate, which was passaged either with or without drug, did not change resistance levels, but passage in the presence of fluconazole resulted in significantly increased tolerance. So when we take these isogenic isolates with different tolerance levels and measure their intracellular fluconazole using the fluorescent probe, we see that the high tolerance ones, as an example here shown in blue, accumulate a lot less fluconazole, whereas the low tolerance isolates tend to accumulate a lot more fluconazole. So this directly illustrates this idea that tolerance inversely correlates with the amount of drug accumulating in isogenic populations. So this means that to some extent, we can use this fluconazole probe as a reporter for azole tolerance. And you can imagine that now that you have established this heterogeneity in the population, you can then sort this population based on either the expression levels of different pathways that contribute to tolerance, or based on how much drug they accumulate intracellularly. And it's likely that cells with high tolerance, because they can still grow in the presence of drug and benefit from extra time and extra cell divisions, will be much more likely to potentially develop true drug resistance. So there's data from a couple of studies now to suggest that this phenomenon is important in the clinic. And we've drawn this conclusion because we see an association between tolerance and the ability of cells isolates to persist in patients during multiple rounds of basal therapy. For example, isolates that are cleared by fluconazole tend to have a lot lower tolerance levels, whereas isolates that persist in infections during multiple rounds of fluconazole therapy have similar MICs but much higher tolerance levels. So importantly, we can detect tolerance in multiple Candida species, as well as in Saccharomyces cerevisiae. So what I'm showing here are Candida tropicalis, Candida Kurzai, Candida glabrata, and Paropsilosis, and their tolerance levels with different azoles, as well as with 5-fluorocytosine. And we can also detect tolerance in response to fungicidal drug. And I'm showing here micafungin, caspofungin, and amphotericin B. Although it's a tolerance, it's much more apparent with fungistatic rather than fungicidal drugs. So for example, we can see similar phenotypes on plates. Echinocandins in particular tend to elicit very diverse drug responses on these E-strip plates. And this figure here illustrates different Candida species that display either trailing growth or paradoxical growth. So trailing is the phenotype that we now call tolerance for azoles. Please note that we need better and more consistent definitions for these phenotypes. And what I want to highlight here is that these phenomena are observed at actually quite high frequency. So over 50% of the isolates that were tested in this study from Candida auris and Candida dubliniensis exhibited either trailing growth or paradoxical growth. And it's important to stress out here that we currently have little to no mechanistic understanding for how these phenotypes arise. So tolerant cells can encompass anywhere between 10 to 90% of the cells in the population. So quite a large subset of the population. However, the other phenomenon I want to mention here is heteroresistance. So heteroresistance is defined as the existence of a subpopulation of cells with significantly higher resistance levels. So typically, this has been defined as at least 8-fold. We can consider a population being heteroresistant as if we find cells with resistance levels that are at least a log higher. So these would be in an otherwise susceptible population. So usually these cells are very, very rare, usually 0.1 to 1% of the population, which makes these variants especially hard to detect because most assays average population behaviors. So we see heteroresistance in Candida albicans as many of the clinical isolates that have been exposed to fluconazole treatment in vivo tend to form large colonies in the zone of inhibition. So these colonies appear completely uninhibited by the presence of the drug. When we take these isolates and we test them, we see that they are much more resistant. And we can see this in MIC assays as well. So from bacterial studies, we know that heteroresistant cells tend to be quite unstable. So we wanted to see whether this was also true for Candida albicans heteroresistant cells. So what happens when we take these colonies and passage them in the absence of drug for multiple generations? So what I'm showing in this plot are MIC50 levels, so resistance levels for parental cells shown in blue, as well as heteroresistant colonies shown in red. And you can clearly see that the heteroresistant colonies are much more resistant than the parental isolate. And when we passage these heteroresistant colonies without drug for more than 100 generations, what we saw was that literally all of them lost the high resistance levels. So heteroresistant colonies passaged in the absence of fluconazole now return to resistance levels that were similar to those of the parental population. So this suggests that heteroresistant cells are unstable, similar to what has been reported for bacterial populations. And I want to highlight here a couple of studies that are quite interesting. And if you haven't followed this topic, I think they're worth reading. So one paper that I want to highlight is a paper in Canada Glabrata, which described heteroresistance as a continuous phenotype. So what I'm showing here are population analysis profiles, or PAP assays, which measure the number of colony forming units that grow on different concentrations of drug. So in this case, fluconazole. So these three plots show examples of a susceptible, a heteroresistant, and a resistant isolates on these assays. And what you can see is that for susceptible isolates, there's a steep decline in the number of colonies that grow at different concentrations of drug. For resistant isolates, basically everything grows, while for heteroresistant isolates, there's a minority of cells, which seem like a lot here, but it's actually less than 0.1% of the population, that grow at 10-fold higher concentration than the MIC of this strain, which was around 8. So these grow at 16, 32, and 64 micrograms per mil fluconazole. So this study also found that heteroresistance is associated with the increased expression of efflux transporter genes, as well as with in vivo persistence. So one way in which these cells can become heteroresistant is to increase expression of transporter genes. So the second study was focused on a different pathogen. So heteroresistance has also been described in more detail in Cryptococcus neoformans and Cryptococcus gattii. And here you can actually see the reference strain for Cryptococcus neoformans, and you can see both tolerance and heteroresistance as phenotypes of these strains in the presence of fluconazole. So you can see small colonies growing in the presence of 60 micrograms per mil fluconazole. This should be micrograms, as well as heteroresistance. So these large uninhibited colonies that grow at high levels of the drug. So what was interesting about the Stone et al. Study was that they found heteroresistance being associated with recalcitrant cryptococcal meningitis. And they also found that heteroresistant cells in general tended to display supernumerary chromosome numbers, supernumerary chromosomes. So in this case, heteroresistant colonies, they all had increased numbers of chromosome 1. So these were disomic rather than monosomic for chromosome 1. This was associated with fluconazole resistance because when these isolates were passaged without drug, they saw a loss in fluconazole resistance correlated with the loss of the disomic chromosome. So this emerged as a mechanism for heteroresistance in this pathogen. So both ERG11 and AFR1 genes are found on chromosome 1. ERG11 is the target of fluconazole, while AFR1 encodes a drug efflux pumps. So this resulted in increased efflux levels, which positively correlated with the high level of heteroresistance. And this really means that aneuploidy and increased efflux are kind of emerging as key mechanisms for how these cells become heteroresistant. So fortunately, there's some indication that combination therapy of 5-fluorocytosine and fluconazole can prevent the emergence of heteroresistance in the species. So that's something that's worth examining in other species as well, whether combination therapies might prevent some of these phenotypes. So one key take-home message is that both tolerance and heteroresistance are associated with persistent infections in multiple pathogenic species. So studying antifungal drug responses is likely very important for the managing of these infections. But it's also very scientifically stimulating given the diversity of mechanisms that these cells might employ to actually escape drugs. So to do this, we need better tools. We need to do more single-cell analyses rather than averaging populations. And we need to understand what are the different routes that cells might take to deal with drug exposure, as well as whether tolerance and heteroresistance might be intermediate stops on their way to developing true resistance. So I want to thank members of our team at the Pasteur and at Brown for contributing to this work, and in particular Cécile Gauthier and Thomas Murphy, who have spearheaded some of these work, as well as our wonderful collaborators for all of their insights and support, and in particular to Judy Bourbon, who's really been driving the field in a lot of these directions. And you'll notice that she's a an author on a lot of the papers that I talked about today. And I'm happy to take any questions.
Great, thanks, Jules. So Oliver, do you want to kick off the Q&A section?
Sure, yeah. Thank you very much to both speakers. Really excellent presentations. And we can also see this because a lot of questions already pop in. And I'll start with questions for Duncan. And the first question is on the specificity of zinc or other metal ion transporters. How specific are they? Do they selectively transport zinc, or can they also transport other ions?
Okay. Yes, so there are two types of zinc transporter. The ZRT related ones transport from non-cytoplasm to cytoplasm. So that's the two cellular uptake systems, and also intracellular organelle export systems. That's one class, non-cytoplasm to cytoplasm. The second class is in the opposite direction. So cellular exporter or intracellular organelle importer. So all of the cellular importers in Candida we've looked at are zinc specific. And intracellular ones are zinc specific. One of the ones that does it in the opposite direction seems to transport manganese. But it certainly is possible because orthologs in plants can, orthologs of those ZRT1, ZRT2 system that I talked about there, which was zinc specific, orthologs of that in distant related orthologs of that in plants can transport iron. So you can have relatively subtle changes in the transmembrane helices, and that can alter metal specificity. So yeah, it's possible, but in Candida, these seem to be mainly zinc specific.
Okay, great. And then another question that's also an interesting one. What is the mechanism that causes the PRA1 to return to the surface of Candida albicans once it's been secreted?
Yes, that's a good question. And I mean, when we do experiments with like kind of high concentrations of PRA1, we get it to reassociate in the Eppendorf or whatever. In reality, that secreted PRA1 is likely being secreted out probably quite a short distance, right? Perhaps within the invasion pocket that Jules Ene and Bernie have talked about, and chelating zinc, perhaps even within the cell wall. So although the cartoon shows like out and back, most of the reality is probably quite a short distance zinc capture. Now, if we knock out the C-terminus of PRA1 with those cysteine histidine motifs, we prevent reassociation of PRA1 to the cell. And if we knock out the cysteine histidine motifs of ZRT1, the same happens. So what we think is happening is the cysteine histidine motif is binding zinc, and then we've got the three cysteine histidine motifs on the ZRT1 receptor. And it's probably initially a zinc bridge interaction between, right, allowing them to come together. And then the fact that you've got three on the receptor, only one in the zincophore, of course, that could be heterodimerization. We think that allows stronger chelation by the receptor. And of course, the receptor is also a transporter, which pulls it off into the cell. And we think that allows release of PRA1 and potential recycling of the zincophore. But that's the model.
Okay, great. And I'll do one other question, and this is related to the story you talked about histoplasma and cocci with the different adaptation to environmental conditions. Do you see similar patterns of loss or gain of PRA1 homologs in environmental fungi?
It's difficult to tell actually, because when you look at, I mean, I've got databases, we know like the pH of the world, right? And in some continents it's really granular, like readings every mile. But it's quite difficult to nail down because you find these ITS studies, right? And it gives you OTUs and stuff, and it says, well, most of the species are ascomycete and the rest are basidiomycete. And it's like, how this is useless information when you can have Candida tropicalis, which has got the PRA1 ortholog, and Candida parapsilosis, which has completely lost the gene. We really much better. We need to be able to identify way down to the species level in order to answer that, and that's actually at the moment quite difficult to find. The only example I can think of is that the Aspergillus fumigatus. By the way, the Aspergillus fumigatus ortholog of PRA1 is called ASPF2. And Jose Calera has also shown that to be zinc neutralkaline, upregulated, anti-essential for growth under zinc limitation, neutralkaline pH. That gene is conserved within the Aspergilli, most of them, but it has been lost by Niger and one other one, which slips my mind at the moment. And Niger is used in the industrial production of acids. So although there's, it's difficult to find genuine environmental fungi, although I would argue that coccidioides and histoplasma are, for the most part, environmental fungi, there does seem to be quite a few interesting correlations between pH and loss. As I mentioned, Cryptococcus, right, in bird guano, that's acidic.
Okay, great. Thank you, Jay. Over to you.
Great, thanks, Oliver. Jules, there's a couple of related questions here. So first has the concentration of fluorescently labeled fluconazole been compared between susceptible and intrinsically resistant species like albicans versus glabrata? And can you visibly see a lower concentration of drug in glabrata, confirming that the resistance is due to efflux pumps removing drug from the cells?
Yeah, good question. So we haven't yet looked at other species with this probe, but what I can tell you is that the correlation, once you start using different genetic backgrounds, starts to be a bit shaky between how much tolerance these isolates have and the amount of fluconazole they accumulate. And this is in particular related to what are the mechanisms that make those strains be more resistant. So for example, for aneuploid cells, we start losing some of this correlation. So it's not as easy as I tried to talk about it today. Yeah. But it would be interesting to look at Candida glabrata as well.
Great. And in a related question, are there mechanisms of transient or heteroresistance that don't involve lower intracellular drug levels? Any evidence for epimutants beyond mucor, for example?
Yeah, so I haven't seen anything suggesting this, but they could very well be out there, but so far there are very few studies on heteroresistance in fungi.
And I was a little bit surprised, this is a question for me, you know, the duplication of chromosome 1, I mean, genetically, is that more efficient than just upregulating transcriptional regulation of those genes? I mean, it seems to me that the fungus is going through a lot to completely duplicate a chromosome. Is that a more efficient or less energy?
Yeah, I guess we have to think about what happens when cells in general are exposed to fluconazole. And we know from a lot of Judy Berman's work and from others that this is a high level of genetic stress that leads to increased rates of LOH, aneuploidy. So if you think about it that way, it might be faster in terms of number of cells in the population that you might get some of these odd cells that are aneuploid, which then have increased resistance. So we suspect that part of their instability is the fact that they're aneuploid, and aneuploid cells tend in general to have to be associated with intracellular toxicity, and actually Hung Ji-sai has done a lot of work on this. They tend to be associated with fitness costs that wouldn't allow them to be maintained in the population once the drug is removed. Yeah, we haven't done the experiments to see what's the most efficient route to become heteroresistant, but yeah, it's a good thing to think about.
Let me ask one last question. We'll turn it back over to Oliver. What is your view about filamentous fungi and how to address all these questions about tolerance and heteroresistance in filamentous fungi, which are not single cells? And should we sequence single asexual spores, for example, to get at this issue?
Yeah, I saw that question and I honestly don't know because I'm not familiar with a lot of the assays that are used for filamentous fungi. I think sequencing might give you some answers, but yeah, I don't know, to be honest.
Okay, great. Thanks.
I noticed that there are a lot of papers that suggest that these phenomena are also present in filamentous fungi, and this further complicates our understanding of the mechanisms as well.
Great. Over to you all.
Right, Duncan, so back to us, and I have a question here about whether you ever imaged PRA1 in an infection setting and whether it can actually also occur inside a host cell.
Yeah, we're trying to do that. It's tricky though, because when we infect the epithelium, we're working in epithelia now rather than endothelia. But when you infect the host cell with that M-cherry tagged PRA1, there's a lot of PRA1 at the fungal cell surface. And we know from, when I work with Bernie, that invasion pocket, there'll be a lot of PRA1 accumulating in that very tight space. So we might be able to, resolve that. Well, we're trying to resolve that with transmission electron microscopy. But of course, in the cell culture infection model, you've got that very strong M-cherry signal surrounding the fungus. Whatever individual molecules have made it out into the host cell as a whole, we think is possible and likely. But by the time you turn up the intensity of the microscope to see those, yeah, the fungus turns to the sun. So yeah, we're trying to look at that, right? But it's not quite as straightforward. And also we're trying to add the purified PRA1 to the epithelium and see if it's taken, wash and see if it's taken up, because then we can increase intensity, or even stain with antibody if we don't have the fungus itself. So yeah, we're doing it, but yeah, not quite as straightforward as.
Okay. And I have another question here, and this is about Candida glabrata, and the question is, why is it so different? Why has it lost the PRA1 gene? Especially because it's very similar to Candida albicans with regard to niches.
Yeah, well, I mean, in terms of the pathobiology, I'd say maybe not. I mean, albicans gets, let's say, when it comes to an infection and albicans get swallowed by a macrophage, it bursts out. And whereas glabrata lives in the macrophage for days and days and days. But of course, I mean, neither was evolving as a systemic pathogen 100 years ago, I would imagine. So if they are commensals, they're in the healthy status, right? And maybe they occupy slightly different parts of the mouth and the gut and stuff where the pH is a bit different, or rather doesn't fluctuate as much. And remember while the PRA1 deletion mutant and the ZRT1 deletion mutant, by the way, it was unable to capitalize on host zinc when it was infecting the host cell. I go back to the very beginning of my talk where I just took the ZRT1 or the ZRT2 deletion mutant and put them into just culture media, right? Then they were, you didn't need one or the other. I mean, they were redundant at neutrality. So you could quite easily, as long as there's sufficient zinc, you could quite easily lose the PRA1 system and the ZRT1 system and be absolutely fine. So it could be that glabrata lost it during its commensal, or wherever it's been for the last million years, and once it's gone, it's gone. But what I would argue is that when it comes to the pathogenicity, that the presence and the absence of PRA1 may influence the pathogenic lifestyle of different species. Okay, and one other question, this is about the pH optimum of PRA1. You showed that it's regulated and it has this optimum at neutral pH or above 6.2. Is this the same for all homologs in all fungal species? I mean, difficult to answer probably. Yeah, well, I mean, I'm just back from a holiday this week, so that might have been done already in terms of the biochemistry assays. What I can say is that the gene expression measurements that we've made and others have made, right? I mean, PRA1 has been known pH regulated antigen since the 90s. It's regulated in this quite specific way, peaking up at neutrality. And, that mode of regulation is conserved in Aspergillus fluviatus from the cholera lab, right? And they're regulated in the same way. So their regulation is via ZAP1 in Candida, ZAF-A, the ZAP1 ortholog in Aspergillus, which regulate the allele in the same way in response to zinc, and RIM101 in Candida, and PAC-C, the RIM101 ortholog in Aspergillus, regulate the locus in response to pH in the same way. So the regulation is conserved in two representative species of two major subphyla of the Ascomycetes. And then in terms of biochemistry, we either know already but haven't been told, or we'll know soon. At least with the zincophore domains, we can easily synthesize that C terminus. In fact, before I went on holiday, it was already ordered, so it might be there, and we can quite easily do the zinc binding assay with the zincophore domain from any fungus we want, right? And it probably, my guess is it probably will, because the histidine residues are conserved, and histidine has got the same pKa value since since the beginning of time, right? So my guess is that the biochemistry will be conserved, and we know that the regulation is conserved.
Okay, great. And we switch again.
Great. So I'm going to start with a couple clinical related questions in the chat Q&A session here. So one question was, could some clinical treatment failures just reflect really severe immunocompromise of the host, and that maybe an antifungal in and of itself is just not enough, right?
Yeah, absolutely. So that's one thing that we have to work on, entangling the two fungal specific factors that would lead to persistent infections versus host specific factors. So in our study, what we tried to do was to match the two groups of isolates that were either persistent or non-persistent to try to get around this issue. But yes, absolutely.
And then the other clinically related question was this issue of, are we ready yet to consider heteroresistance as a clinical decision point yet? In terms of either considering combination therapy or actually switching a class of antifungal.
Yeah, so given that we don't really know what the mechanism is behind these phenomena, I don't think we are ready. So for example, for bacterial cells, where the mechanisms are much more known, it's still not taken into a clinical viewpoint. It's still, these phenotypes are still not considered when treatments are prescribed. So one way that people postulate that we can get around this is to use combinatorial therapy to try to hit those mechanisms that would enable the formation of either tolerant or heteroresistant cells. So for Cryptococcus in the study that I highlighted, it was very nice to see that combination of 5-fluorocytosine and fluconazole was able to prevent the high frequency of heteroresistant cell development. But I imagine cells, given how flexible they are genetically and epigenetically, will probably find a way around this as well. For tolerance, what we saw was that, again, combination therapy with different inhibitors of specific pathways that enable tolerance was able to clear tolerance, which was quite interesting. But yes, we're not ready. Yes.
And are these phenomena consistent across different culture conditions? Solid, static liquid versus shaking liquid. Like, as we develop potentially clinically relevant assays, is that an important point to consider?
It's definitely an important point to consider. So I would say that the phenomena are consistent, but the degree to which you observe them differs quite a lot. So if you switch the media, if you plate, or if you grow them in liquid, you're going to get a different extent, a different dynamic range for these phenotypes. Yeah, definitely pH, temperature, everything will matter, as we know with Candida.
And how many generations does it take for high tolerance to evolve?
So in some of the isolates that we've looked at, we've seen high tolerance being developed in as little as 20 generations. But we waited a bit longer to get to a higher difference. So we passaged for about 100 generations.
Okay.
And it also depends on how much drug you're hitting them with, whether you're increasing the concentration of drug. So everything will matter.
Okay. Oliver, I'll turn it back.
Right, Duncan, I have a question regarding host response, and as you described how pretty much all of these species have zinc transporters and zinc transport proteins. Is there an immune response against these proteins and selection pressure?
Yeah, and I think that's one of the reasons that loss of the praluent zinc flora in the environment precedes the evolution of most human fungal pathogens. So it's quite a clever system. That, I mean, it's been described in the bacterial world. You get nutritional immunity and innate immunity. They integrate, right? If you think about the immunogenicity of heat shock proteins, which are switched on by fever, it's quite clever that nutritional immunity forces the bug to switch on PRA1 if it has the gene, and it simultaneously evolved to target PRA1. So a really nice study 10 years ago now, showing that PRA1 can be targeted by neutrophils, and we're following this up. So PRA1 is targeted by neutrophils. Candida albicans PRA1 is targeted by neutrophils, and Candida albicans PRA1. If you look at the work from Peter Zipfel, right, your ex-neighbor Oliver, I mean, PRA1 interacts with so many components of the the mammalian complement system, and he's got a more recent paper showing that the Aspergillus ortholog of PRA1 ASPF2 also interacts. So although mammalian immunity may have evolved to target Candida albicans PRA1 or PRA1 in general, I mean, they're conserved and it probably cross-reacts. So absolutely, PRA1 is immunogenic, and this is my hypothesis, right? That albicans and fumigatus are relatively unique human fungal pathogens that exhibit extremely aggressive hypho-mediated growth. They've kept the zincophore. They don't care. Once they get going, they use it to get zinc. They provoke inflammation, and yeah, they don't care. Everybody else, Histoplasma, Cryptococcus, Candida glabrata, etc., has lost the gene. I mean, millions of years ago, it's lost the gene, and then when it infects the host, it has that slight advantage of at least not getting cleared by neutrophils during, via that mechanism. Of course, there's beta glucans and everything else, but that might be the small the small foothold it needs to not provoke a PRA1 mediated neutrophil response. And yeah, that's a good question. That's one of the things we're actively looking at now.
Right. So, and I'm switching back and forth, and sorry for that, back to ecology. And this is Joe Heidman pointing to the fact that both Kolls and Histoplasma have been identified as species complexes. So there are now new species identified in these former species. And is there a difference probably, or perhaps with regard to zinc uptake, that could also explain ecological differences?
Well, that's a good point, but no, I've not found PRA1 orthologs in any of the Histoplasma capsulatum genomes, right? Whether they're from North, South America or Africa, the gene's gone. And then in Blastomyces, which is the next one out, it's there, but it's broken from its receptor, and the locus is present. Whether the PRA1 ortholog is present in both Kolls immitis and Possidacea, I think it's called.
Okay.
I mean, particularly with Blastomyces, yes, maybe in a million years, right? So you've got Histoplasma in the east and the southwest, which is actually like east of central in America on the acidic soil. You've got Kolls in the alkaline, right? And by the way, I don't know if I mentioned that recent outbreak that Tom Chillers wrote about not that long ago in Washington State, up in the north. It's half acidic, and the outbreak happened to the east in the alkaline regions, right? So it's really acidic alkaline when it comes to not having the PRA1, having the PRA1 locus. Blastomyces, where it's got it but it's broken, kind of like straddles the pH line. If you look at that pH line, so yeah, it could be that Blastomyces it's on its way out. It could be that the loss of synteny could be the first stage in PRA1 loss, because you lose that tight co-regulation, things start to go awry, and yeah.
Okay, and then a question with regard to, let's call it, efficiency of zinc uptake. So is the amount of zinc that can be taken up by an organism related to the mechanism or the number of transporters that is present? Or also, I remember you showing that the binding of PRA1 to ZRT1 has been replaced by a GPI anchor, and could this alter efficiency of the mechanism?
Yeah, I don't really know what's going on with the Saccharomyces one. It is a bit weird, and although it's named ZPS for zinc pH, that's based on large-scale studies, and it's not been functionally characterized yet. In terms of the zinc transporter copy number, I would say yes, probably, but it will really depend on the condition, right? So albicans I thought albicans was like a kind of archetypal Candida or fungus with its two ZRT1, ZRT2 zincophore system, right? Parapsilosis has lost PRA1, right? Which might fit with the fact that it's quite often in skin, which is slightly acidic. But it's duplicated ZRT2, and then that duplicated ZRT2 has duplicated. So it's got 3 copies of ZRT2, and yet it is not as efficient as albicans at growing under zinc limitation at either neutral pH or acidic pH. So it might be we just haven't found the environment yet. I mean, we're using Y and B media with glucose, and then we alter the zinc and we can buffer it or whatever, right? It could be that there's a zinc binding molecule which is present in skin, right? Like psoriasis, for example, that parapsilosis, that duplicated version of ZRT2 in parapsilosis is really good at getting. Of course, this is a lot of work for the future, because we need to take, I mean, ideally every fungal species on the planet, define its zinc uptake repertoire, and then knock them all out, and then do growth assays with every kind of imaginable zinc source we could think of at each pH value. So yeah, it's dead interesting, but it's too much work to have all done already, I'm afraid.
Okay, at least you have a plan. Thank you very much, and Jay, back to you.
Great, thanks. Jules, we have a question about within albicans, are there clades that are more or less prone to display tolerance, heteroresistance to antifungals than others?
Yeah, so we haven't addressed this question specifically. We have tested isolates from different clades, and we don't see an association. So this is not surprising, given that there's literally no association between different phenotypes in Canada and different clades, except for maybe Candida africana, which might be a different species altogether. So yeah, no, nothing that stands out right now.
And then do heteroresistant strains have a higher tolerance by considering fraction of growth within inhibition zone along in that colonies per se?
Yeah, so this is a good question, because one way in which we measure tolerance is using this fraction of growth inside the area of inhibition. So you can imagine that if you have a lot of these heteroresistant colonies growing, it will definitely impact this fog. So this is where it gets complicated, because some of these isolates tend to have both. So if you have an isolate that has both, you will confound your measurements of either heteroresistance and tolerance. So we've been moving away from using that type of measurement when we're looking at heteroresistance, just because it would affect our measurements of tolerance.
And then an interesting question about fluconazole Cy5 localized to the mitochondria in the in the paper, right? So is there do you think this is a real thing, this fluconazole localized to the mitochondria, and if so, is that important in its mechanism of action?
Yeah, so that's a great question. So I think the paper actually hypothesized that because both probes went to the mitochondria, it's likely that all azoles go first and localize first in the mitochondria and then to the ER. But it's also likely that the effects that we see could be off-target effects. For example, ERG11 requires heme synthesis, which happens in the mitochondria. And also there are several reports now that link mitochondrial function, respiration with effects on azole resistance. So there's a link there as well.
Great, thank you. I guess we have a couple of minutes. If there's a couple, do you want to handle a couple more questions, Oliver?
Okay, we'll give it a try. I don't know whether we can finish all of them, but I have a question here, Duncan, with regard to regulation of the zinc transporters. Do you know anything about regulatory mechanisms? The question is specific for upstream ORs or antisense transcripts and their role.
Yeah, not that is known in fungi as far as I'm aware, and there is quite active work in Saccharomyces cerevisiae and Pombe as well. I'm not aware of that in fungi yet. There is one cool example in Leishmania, which is one of these kind of odd species that doesn't really do promoters. Doesn't really turn gene expression on and off like Candida would, switching on when it's needed. And it's a major zinc importer, which obviously brings zinc into the cell. You don't want to bring in too much zinc if there's too much, because it's toxic. And the way that's regulated transcriptionally is that the messenger RNA of the zinc importer is able to bind to zinc within the cell. And when that happens, it's targeted for degradation. So there are kind of RNA level or transcript level examples out there in in the field of parasitology, but not in fungi that I'm aware of.
Okay, so now I have it at 6:30. I think we can stop here. We should take the last seconds to thank the two speakers for really great talks and also the audience for vivid discussion. I remember everyone to the next MycoTalks event, which will be on August 28th. And with that, I think we can conclude the session. Thank you very much.
Thank you.
Thanks. Bye-bye.
Bye.